Hey, Lane.
Hello.
Anastasia. Good to see you. Here comes Vita. Savannah. Hey, Ben. And Arwa. Everybody's rolling in. Are you ready for a break? I think this time of year, everybody's ready. Okay, that's fine. That's fine. All right, terrific. If everybody who is able to can turn their cameras on.
There was no negotiation. We're not
We
gonna
have to start out with the cameras on. If you have a reason that you can't, let me know. That you can't leave it on, let me know, but otherwise I prefer them to be on. Terrific. Great. Helps me get to know you, helps you get to know one another. So I think that's great. All right. So Abe, I had no idea that Hannah was your sister until you appeared in the camera the other day when she was in class. I was like, oh, there's Hannah. Hey, I didn't know you guys were siblings. Some of you I know have siblings that are in my other classes, but a lot of times I don't pay so much attention to last names, so I wouldn't necessarily make that connection. So, yep. All right. Anybody have any questions before we get started? Okay, so we do not have class next week. You have class still tomorrow and Thursday. You have lessons. I'm sorry, tomorrow and Friday. You have lessons that are due, and then you've got all next week off. So if you are a little bit behind on the due dates, make sure that you use next week to get caught up, because at the end of the semester, you only have 1 week after the semester ends. So our semester ends on December 20th. That means December 27th is the last day that you can turn in first semester work. All right, so use the time to get caught up. Keep in mind, though, that I'm on vacation too, so I won't be grading next week, at least not very much. And really, it depends on whether or not my grandson is born. I have a new grandson that's due any day now, so we're waiting for him to come into the world. And if he comes soon, next week, then I will definitely not be doing a whole lot of work. I'll be taking care. Yeah, it is very exciting. Thank you, guys. It is awesome. New little life forms, right? All right. If nobody has any questions, I'm going to go ahead and get us started today. We actually have really a lot to get through. So we are going to be, let me know if you can't see that. Okay. We are going to be talking about how we power life. So we've been learning about all these things that the body does. We've been learning about the different systems of the body. Today, I want to just take a step back inside the cell and talk about how the body gets the energy to do all the things that it does. So what is the name of the molecule that gives us the energy that we need? All living things use this molecule for energy. What is it? Yep, ATP, Olivia. What does ATP stand for? Anybody? There you go. Monica, great. Adenosine triphosphate. So you guys learned about ATP, of course, back at the end of October. There was a lesson in the muscle unit that was called energy sources, but I want to go a little bit deeper today, give you more information that you got. Now, this topic is pretty complex. I actually think that the idea of making energy, the cellular respiration, is one of the most complex topics in all of biology. So I'm going to move quickly because we've got a lot to cover in just an hour. As always, if you want more detail or you don't understand what I'm talking about, stop me. Do your best today to stay connected the whole way through, because if you zone out for a minute, it might be a little more difficult to jump back in. All right. I know that's a little bit hard when it's right around lunchtime for most of you, but do your best. So ATP stands for adenosine triphosphate. Now, in order to really understand ATP and how it works, how it does what it does, we have to understand enzymes. Anybody know what an enzyme is? What's a good way to describe an enzyme? Yep, good, Anastasia. It catalyzes chemical reactions. So what does that mean? What does catalyzing a chemical reaction mean? Speeds them up, exactly. A catalyst is usually, an enzyme is usually going to be a protein. It is going to be something that catalyzes reactions. It speeds them up. So that's what catalyst means, something that speeds up the chemical reaction. But what's really important about this catalyst, these enzymes, is that they are not permanently changed or used up in the process. They can be used over and over and over again. And that's really important when we're talking about ATP. So when we are talking about an enzyme, they are specific for what they will speed up. That means if they speed up the reaction that breaks sugar down into, like a complex sugar down into simple sugars, that's all they do. You don't have an enzyme that is multi-purpose, that does a lot of different kinds of reactions. They are specific. The reactants are what we call substrates. There's one enzyme for every different substrate that we have. A substrate is just a molecule that the enzyme acts on, and these enzymes are reusable. Now, this is important. They almost always end in ASE. So whenever you see ASE at the end of a word, that usually means enzyme. Examples are amylase. Amylase is made by your salivary glands. Sucrase breaks down sucrose or sugar, table sugar. Lactase, this breaks down the proteins that are found in milk. So people who are lactose intolerant often don't have lactase, or they don't make enough of it. So they sometimes can take lactase by pill. Are any of you guys lactose intolerant or know somebody who is? So there's certain milks that you can buy that have lactase in them, or you can take pills or powder before you have a meal in order to get that lactase to break down the lactose. And maltase breaks down maltose, which is another kind of sugar. Savannah, you developed it a few months ago. It's not as hard to be lactose intolerant as it used to be, because there are a lot of things that you can do now in order to undo the damage that might be done. And here's the good news. Just like you developed it a couple months ago, you could also grow out of it. Some people grow out of lactose intolerance. So hopefully you're one of them. All right, no, so how do they work? Enzymes work by weakening the bonds between the atoms that make up molecules. So that lowers the activation energy. You can think of it this way. Think about if you are in a group that's going on a hike and you come to a hill, a big hill. You've got to climb up that hill. Somehow you have to get to the other side of the hill. That's your goal. Get to the other side of the hill. Well, without help, it would take a lot of work and a long time for everybody to climb up that hill. Now imagine that there's a tunnel right through that same hill. That tunnel is like an enzyme. It's going to provide a shortcut for you to get to the other side. That's what an enzyme does. It provides a shortcut. And just like the tunnel is there for everyone to use forever, the enzyme is there to be used for a really, really long time. The enzymes are not used up in the process. So here we can see how that happens. This would be what the blue would be without an enzyme and the black would be with an enzyme. So right there, there has to be a certain amount of energy put into any chemical reaction in order to get it started. If we have an enzyme, though, it takes much less. So much less. We lower the activation energy that is needed in order for the chemical reaction to take place. Now, this is what an enzyme looks like. That's like an artist's representation of an enzyme. Each of these little spots would be a different kind of an atom. We use these little pictures to give us an idea. So sometimes an enzyme will take one substrate and break it into two. Sometimes it'll take two substrates and combine them into one. So how do they work? Enzymes work by weakening bonds. So at the top here, we have a starch molecule, like you would have in bread or cracker or pasta. Those are made of starch. You got to break them down in order to get the individual molecules. That happens in the process of digestion. Now, enzymes make these bonds in between all of these molecules weaker. Once they're weaker, now you don't have to take much energy to break them down. You know, it's like when you're having an arm wrestling fight with somebody and you tickle them. Now you can win really easily because they've lost their energy, they've weakened. And the same thing happens with an enzyme. We have the enzyme get right in here and weakens that bond in between the two. Remember, whenever we've got any kind of chemical reaction, what goes in to the reaction is called a reactant. What comes out is called a product. So reactants react to produce the product. Okay, reactants react. Now, in an enzyme reaction, the reactant is called a substrate. So where is the reaction in this process? The reaction, the enzyme, I'm sorry, where's the enzyme in this process here? The enzyme is actually going to bind to these reactants, and that will help to produce the product more quickly, like this. Remember, the reactants are called substrates. The enzyme binds to the substrate and makes the product happen. The whole thing altogether is what we call an enzyme substrate complex. So you have your substrate, that would be the reactants, the things that go in. It's going to join to a special place on the enzyme, and the whole thing altogether is called an enzyme substrate complex. Now, any enzyme is going to have a place on it where the enzyme is going to bind to the substrate, and that's what we call the active site right there. When that happens, it is changed chemically. That substrate, either it's broken apart or it's joined together. It's in some way changed chemically. The active site itself makes the enzyme change shape. We call this induced fit. So two reactants can become one can become two or more. And now keep in mind, whenever we're talking about an enzyme, this chemical reaction would happen anyway. Enzymes don't make things happen, they just speed them up. Without the enzyme, the reaction would still occur, but it wouldn't happen fast enough for life to exist. So these enzymes make things happen fast enough that it can be compatible with life. So they're pretty important. Cells are filled with enzymes. There are thousands of enzymes in every cell. They're all different shapes, if you will. They're all different chemical shapes, depending on what they do. You have lots of them in your digestive system. So this is an example right here of an enzyme. So the purple is the enzyme. The active site would be these sort of geometric shapes right here. So here you have sucrose, that's table sugar. Sucrose comes in and it is bonded to the enzyme and broken apart into glucose and fructose. So sucrase is the enzyme that breaks down sucrose into glucose and fructose. And that happens in your mouth. Your salivary glands make the amylase that contains the enzymes that are necessary to break down a lot of the food that you eat, your carbohydrates. Carbohydrate production begins in the mouth. If you're a person who bolts down your food, who doesn't chew your food very well, you're not making the best use of the carbohydrates that you take in. You need to be able to chew them to mix them with that amylase. So amylase breaks molecules apart, just like this. We start with our starch. Amylase is going to break them down into smaller molecules. So when you eat something like a cracker or anything else that's starchy, that saliva is going to mix with your food. And the amylase is going to start breaking starch down into maltose molecules. You can actually do a fun experiment and prove this to yourself. Maltose tastes sweet, but starch does not. So the starch in a cracker doesn't taste sweet. But if you put a cracker in your mouth and you chew and chew and chew and chew without swallowing, you just keep mixing it with your saliva, it will start to taste sweet. So try that sometime. If you've got a cracker in your house, give it a try. Now, when we study the digestive system, we're going to learn a lot more about enzymes when we study there. But let's focus on ATP and how enzymes are necessary for those. So like we said, ATP stands for adenosine triphosphate. So we're right in the middle of the muscular system. Learning about that, muscle cells use a lot of ATP. In fact, a working muscle cell uses about 10 million molecules of ATP every second. That's one muscle cell. One muscle cell uses 10 million molecules of ATP every second. And surprisingly, muscle cells use the second largest amount of ATP in the body. Anybody want to take a guess at what system uses the most? Muscular system uses the second most. Which system uses more ATP than muscles do? No guesses? Respiratory system's a good guess, but no. Does seem like it because we're constantly breathing. Yeah, nervous system. The nervous system uses the most. The nervous system actually uses about 25%, 1/4 of all the ATP that we use, that we generate. So it's a lot. But muscles use a lot as well. And here's what they use it for. They use it to move things against the concentration gradient. Remember way back when we learned about movement of things across the cell membrane? We learned that there's passive transport where things just kind of move on their own. And then there's active transport. That takes ATP. That moves things from where they're not very concentrated to where they're already really concentrated. That takes energy. That whole process of mitosis takes energy for one cell to become two. The movement of a cell. So some cells move by flagella or with cilia. You've got cilia lining all of the cells that are in your respiratory tract, in the top of your respiratory tract. Inside your nose, you got little hairs. You can see the hairs. But if you could look at the individual cells, you'd see that they have little cilia. And that cilia has to move in order to beat stuff out that would get down into your lungs otherwise. Production, transport, and storage of proteins, and really everything that's done in the body uses energy. So where does that energy come from? How does the energy get into the ATP to begin with? Where do we get it? Yep, from food. Now, you guys probably have known since kindergarten, because for some reason, kindergartners always remember photosynthesis. That's a term that you probably know most of your life. So you've probably known for a long time that plants use energy from the sun to make sugar in the process of photosynthesis. Now, the sugar that they make is called glucose. And when you eat the plant, you get that glucose. Or you eat an animal that ate a plant, so you still get the glucose. But the problem is you can't use glucose. Even the plant can't use glucose, right? The plant makes energy, but it can't use that glucose that it stored the energy in. It's locked into the glucose molecule. So we have to find a way to unlock the energy that's in that glucose molecule so that we can use it. Plants do too. They have to have a way to get that energy out of the glucose molecule. So the energy gets out of the food and into ATP through a series of chemical reactions. They take place partly in the cytoplasm, but they also take place in that organelle that is the energy producer of the cell. Anybody remember what the name of that organelle is? The one that makes energy, that produces ATP. Mitochondria. Yep, it is the mitochondria that are the energy producers of the cell. So the energy, think of the energy in a glucose molecule as like a giant water container, like a big one like this. Well, that's a ton of water in there, but it's not really useful to us if we just want to drink. You can't really take a drink and pour that all over yourself. But if you could put that energy or that water from that big water container into 40 little water bottles, now those little water bottles are really useful. So the little water bottles are the ATP, the big container, that would be the glucose. So it is a series of chemical reactions that does that takes it from the glucose into the ATPs. Now look at the two equations. This is photosynthesis right here. We start with carbon dioxide. And water. So 6 carbon dioxides and 6 waters will yield, in the presence of sunlight, glucose and 6 oxygens. Now look at the other one. Look at the respiration or cellular respiration on the bottom. Glucose and oxygen, carbon dioxide and water, carbon dioxide and water. They're opposites of one another. The products of one are the reactants of the other, and vice versa. So photosynthesis uses energy to build glucose. Cellular respiration breaks down those glucose molecules to create adenosine triphosphate molecules. So why do we have to do this? Why is it beneficial for cells to use ATP? Why do you think the body doesn't just break the bonds of the glucose molecules and let the energy out and use it that way? Actually, it would just be too much energy. Yeah, too much energy to use right away. You're right. Absolutely. Just like drinking from that big 50-gallon drum of water would be too much water. It flood over you. So you use smaller thermoses to get the water. The same way with ATP. It would actually destroy the cell. If you put all the energy that's in a glucose molecule, if you just let it loose into the cell, it would destroy the cell. It would be too much. It would burn it up. It would be like electrocuting the cell, so to speak. All right, so let's take a look at an ATP molecule. Remember that adenine is the nitrogenous base that we use. We've got 4 nitrogenous bases: adenine, guanine, cytosine, thymine, and then uracil as well. But in an ATP molecule, we use the one called adenine. Ribose is the sugar. And look at these 3 phosphate groups. It has phosphorus and oxygen. This is a phosphate group, this is one, and this is one. That's what an ATP molecule looks like. Now, these bonds in between here, they hold a lot of energy. When a phosphate is broken off right here, the energy that comes out is a lot. That's the bond that breaks to release the energy. Whenever any part of the cell needs energy, it breaks off one of those phosphates. So the question then becomes, well, how does the phosphate get put back together in the first place to be able to use it? Well, when ATP reacts with water in what's called a hydrolysis reaction, if you've had chemistry, you know hydrolysis reaction is one where something is broken down by water. So when ATP reacts with water, you end up with ADP, adenosine diphosphate, and a phosphate group and a lot of energy. So we got this phosphate group over here. We've got this ADP, but we need millions of these ATPs every second. How do we get that phosphate back on there? How do we add that on so that we can get that energy to use again another day? That's one of the beauties of ATP. It is the ultimate renewable energy source. When the phosphate is taken off, ATP becomes ADP. We have a process inside our cell, partly in the cytoplasm, partly in the mitochondria, to put that phosphate back on. So we take it off, and then we put it back on, and then we take it off, and then we put it back on. We have a lot of ATP inside all of our cells. Every cell in the human body contains about 1 billion ATP molecules. You getting an idea of how tiny all this stuff is? Remember, the nucleus has 6 feet of DNA. Every cell in your body has about 1 billion ATP molecules. Really, really small. Every one of those molecules, those ATPs, is made and broken down and made and broken down somewhere between 1,000 and 1,500 times a day, depending on the cell. Basically, the human body makes and breaks down more than its weight in ATP every single day. So the ATP stores in your body are actually pretty small. Wait a minute, I thought we said there were 1 billion. There are 1 billion. It might sound like a lot, but it's used up really fast. We only actually have enough ATP. That 1 billion molecules of ATP is only enough for a cell to go through maximal work for about 2 seconds. So we have to make it all the time. You've only got enough ATP in every cell to do the full amount of work that cell could do for 2 seconds. So it has to constantly be being produced inside the cell. It is reassembled. That phosphate is put back on the ATP in a process called cellular respiration. All the steps in the process are called cellular respiration. And look at the final result right here. The final result is ATP. Now, this looks like it's just one little reaction, like, oh, we take this glucose and these oxygens and we just rearrange them into carbon dioxide, water, and ATP. We're going to learn it's actually a lot more complicated than that. So let's look at the reactants of cellular respiration. Remember, reactants react, they go in. So we've got sugar and oxygen. Sugar is this glucose C6H12O6 and oxygen. So the products of cellular respiration, what comes out? Carbon dioxide, water, and ATP. Now this carbon dioxide right here, that's really toxic. In fact, if you hold your breath and you start feeling bad, the reason that you're feeling bad isn't because you're lacking oxygen. The reason is because the carbon dioxide is building up. So you have to breathe out in order to get that carbon dioxide out. That's how we get rid of it. We get rid of that carbon dioxide by breathing it out. We'll learn about that in the respiratory system. Understand, when you put the word cellular before this word, it doesn't mean breathing. Now, breathing is necessary for cellular respiration, but cellular respiration means the way ATP is made inside the cell, in the cytoplasm and the mitochondria. All right, are you guys with me? Everybody's still awake? I know I'm going real fast here. Now, there's two different ways that living things can get that energy out of a glucose molecule. I eat my food, I've got to get it out. I've got to get the energy out of the glucose molecule, put it into my ATPs. How do I do that? That really depends on whether there's oxygen present or not. So cellular respiration can occur with oxygen and when there's either not enough or no oxygen in order to have that happen. So we'll talk about both of those. Aerobic means oxygen. When you do aerobic activity, you're breathing deeply, you're taking in a lot of oxygen. So aerobic happens when there's oxygen present. So there are three parts. We talked about these back in October. There are three parts to aerobic respiration: glycolysis, the citric acid cycle, and oxidative phosphorylation. Glycolysis takes place out in the cytoplasm, and that can happen without oxygen. So that part right there doesn't need oxygen in order to happen. So take a look that glycolysis involves an NADH right here. This NADH can bypass the citric acid cycle altogether and go to the electron transport chain. Now, two pyruvates, glucose is broken down into two smaller molecules way out in the cytoplasm. As soon as it gets in there, it's broken down into two pyruvate molecules. Those pyruvates then move into the mitochondria and they go into the citric acid cycle. The pyruvates don't go on to they don't bypass the citric acid. The NADH can. Now, the whole point of this citric acid cycle is to make NADH and FADH2. You don't need to know what those mean for now. If you want to, look them up. But basically what they are is molecules that carry electrons. So they need to carry electrons that came off the glucose and the pyruvates into the electron transport chain. They're going to move into this inner folded membrane of the mitochondria. So this orange, that's the mitochondria. You have your outer membrane, and then you've got your inner folded membrane. So these molecules, this NADH and FADH2, are going to move into this inner membrane to be ready for the electron transport chain. They're carrying the electrons. All right, let's look first at this first part called glycolysis. So if you have enough oxygen or if you don't have enough oxygen, either way, this whole process of making ATP starts with glycolysis out in the cytoplasm, not in the mitochondria. How many ATPs are produced here? Take a look at the picture and tell me. How many are produced? 2, yeah. Actually, if we were to look at the reaction, 4 of them are made, but 2 of them are used up. So it takes some energy in order to fuel glycolysis. It takes 2 ATPs to make glycolysis work, but they make 4, so we have a net of 2. All right. Right here, you can see you start with glucose. It breaks it down into 2 pyruvates. Glucose has 6 carbons. Each pyruvate has 3 carbons. Now, what happens to that pyruvate? That is then going to go into the citric acid cycle. But remember when I said it looks like it's just one reaction, but it's actually a bunch of them, a bunch of series. This is what the reaction for glycolysis looks like. Just look at the amazing complexity. That's the first and simplest stage of cellular respiration. Now, look at all the enzymes. I'm going to circle them here. A lot of enzymes just in the first and simplest step of those. Now, remember, these enzymes are not permanently changed. So they're able to speed up or catalyze these reactions over and over and over again. Given how many ATPs have to be made every second in the cell, that's pretty important. Yep. If there's a malfunction with just one of those enzymes, does that mean the whole reaction won't work? Yes, it does. Now, there are some redundancies. There are some times that, something can take over for something else. When you think of all the possible things that there are that can go wrong with the human body, it really is miraculous that we are all functioning as well as we are. Really take a minute to appreciate your body. Even if it's not doing, even if you got a bad cold, or even if you're lactose intolerant, even if something isn't working well for you, think of all the things that are working well for you. Yep. All right. So hitting it all again, glycolysis takes place in the cytoplasm. It looks like it's one reaction, but it's actually a series of reactions. And once we have this glucose that's been broken down into pyruvate, that's what all this was about. Look, we started with glucose. Those are the reactions that it takes just to get us to 2 pyruvates. All of those reactions happen. So once we have pyruvate, now it depends on whether there's oxygen or not. Now, if there isn't any oxygen, we have a system that's called the phosphagen system. So this is a kind of anaerobic metabolism, a way of using, of producing ATP without enough energy. It uses what's called creatine phosphate, which is where the phosphagen comes from, in order to make ATP. Now, during the short term, when you've got like a lot of activity, like let's say you're running a marathon, large amounts of power need to be produced by your muscles in order to keep you moving in this marathon. So you've got a very high demand for ATP. This phosphagen system is the fastest way to make more ATP. So creatine phosphate, which is, whoop, my circle's off, should be right here. This creatine phosphate right there, that is stored in your skeletal muscles. And that can donate a phosphate right here in order to produce ATP. In the end, we have ATP produced. Now, this is really good at giving you ATP for about 30 seconds of high intensity activity, like sprinting. During the rest, you'd have to take rests in between in order for your ATP to build back up. But this can give you that burst if you're running away from a dog, right? If you're running after your prey, if you're a hunter and you're trying to get some, it can give you that burst that you need. Another way that we can make ATP without oxygen is what's called fermentation. So this is also called anaerobic respiration. And there's 2 kinds of it. There's lactic acid fermentation and alcoholic. Now, alcoholic is not used by people, so we're not going to really even talk about that. That's used by like bacteria in order to make bread. They use alcoholic fermentation. But fermentation takes those 2 pyruvic acid molecules and it regenerates the NADH into NAD+ in order to keep glycolysis going. That's the whole idea. Lactic acid fermentation keeps glycolysis moving, never goes into the mitochondria. This is all happening out in the cytoplasm. Now, one of the problems is that lactic acid will build up in your muscle cells. It will lower the pH of your muscle cells because it's an acid. It makes your muscle cells a little bit more acidic. That acidic environment can mess up the function of the enzymes. They don't work as well in an acidic environment. That's why right after exercise, you're sometimes kind of sore. You're a little bit fatigued. Now, that's only within the first maybe 12 hours after exercise that you feel that because of lactic acid. If you're sore longer than that, it's not because of lactic acid. The lactic acid is cleared pretty quickly from your muscles. If you're sore 24 hours after exercise, you've done some little micro tears in your muscles. And it's not lactic acid. But that lactic acid comes with a price. We keep producing ATP, but we pay the price of having sore muscles and having that little bit of acidity prevent the enzymes from working as well as they should. Now, most of the time, we have enough oxygen. That's why we breathe faster when we run. No, it doesn't appear to have any lasting damage. Nope. Good question. It's cleared very quickly. Your body does not, your brain only wants a very narrow range of acid base. There are lots and lots of systems at play, lots of those negative feedback reactions that will get rid of any acid or get rid of any alkaline that's in the blood. Long as you can deal with the pain, it won't hurt you in the long run. Well, no, that's not true. The lactic acid won't hurt you in the long run, but the muscle fiber tears could. Yeah, that could take a lot longer. And like the whole idea of working through pain, They used to say that. Now you want to work. So it's kind of a balance. Like the little bit of burning in your muscles is a sign that your muscles are working, and that's okay. If you get to the point where you say, okay, this hurts now, this is pain, that's when your body's telling you that you need to back off. You need to rest. You need to allow more ATP to be generated. You need to allow a little bit of rest in between, a day in between different kinds of activities that you do. Yeah, good question. Okay, so most of the time we have all the oxygen that we need. Most of the time, those pyruvates move inside the mitochondria. They move into this part in here that's called the matrix, the mitochondrial matrix. So remember, mitochondria are the energy producers of the cell. So those two pyruvates move into the mitochondria. So right here, this is the cell itself right here. This is the cytoplasm. This square right here is just a representation of the mitochondria. So this outer circle, that's going to be your membrane. And the outer part right here, that's going to be the outer membrane of the mitochondria. This is the inner membrane of the mitochondria. Glycolysis happens out here in the cytoplasm. Those high energy NADH and FADH2s are going to be made in glycolysis, and they're also made in the mitochondrial matrix during the citric acid cycle, sometimes called the Krebs cycle. Hopefully you guys are remembering some of this from when we talked about it in October. Now, the citric acid cycle and the electron transport chain, those both happen inside the mitochondria. So right here in the matrix, that's where the citric acid cycle happens. Those electron carriers, the NADH and the FADH2 that were made in the citric acid cycle, move into this inner folded membrane. Now, in reality, I've got it like stretched out here. In reality, it looks more like this folded in order to increase the surface area. So there's more room for ATP to be made. So right there, you can see it in that twisted inner membrane. So when there's oxygen, the pyruvates, those two pyruvate molecules that were made in glycolysis, they move into the mitochondria, into the citric acid cycle. It's sometimes called the Krebs cycle, two names for the same thing. This cycle right here is so complex. I had a whole semester course in graduate school on this system, right? A whole semester just to learn about the Krebs cycle. It is really complex, really important. All we need to know in this one is that it produces just a little bit of ATP. It doesn't really produce enough to be useful. What it does, what this Krebs cycle really is good at, is making this NAD and this FADH2. That's what it's really good at doing. They are the electron carriers. So why does it matter? Why do we need to carry electrons? We need to carry electrons to what's called the electron transport chain. Remember what electrons are? You got an atom here. You got your nucleus right here with protons and neutrons, and then the electrons orbit around the outside. Now, these electrons have a lot of energy in them. So the ones that are involved in the Krebs cycle are very high energy electrons. They're picked up by NAD, and then it becomes NADH. They're picked up by FAD, and they become FADH2. And then they go into the Krebs cycle. This is a little better representation of the Krebs cycle. Again, look at the enzymes that we have here. This is the Krebs cycle, and this is actually not even the entire Krebs cycle right there. All right, so look also at the blue. Look at the energy-rich electron carriers in blue. NADH, NAD, NADH, NAD becomes NADH, and two hydrogens. There's another one up there. All right, so they're carrying the electrons into the electron transport chain. So where do the electrons go? They go to the inner membrane, this right in here, this folded inner membrane part. Notice the space in between the inner membrane and the outer membrane. Well, high energy electrons are going to pass from the NADH into proteins that are embedded in this inner membrane. This is the inner membrane right here. This is the outer membrane right there. We're looking at this blown up. They're going to be passed to these proteins that are in that inner membrane. and then they're going to be moved along from one to the next, to the next, to the next. And as they do, energy from them pushes hydrogen ions into this space right here. 4 hydrogens, 4 hydrogens, 2 hydrogens, and then where are all those hydrogens going to go? There's a little bit of space right here. There's not a lot. You got hydrogens being pumped in at each one of these little proteins along the way. They're going to build up over there. Now, as those electrons go into those proteins, are absorbed by those proteins, they actually also are going to leave a lot of their energy inside this membrane. One side of the membrane is going to be positively charged and the other side is going to be negatively charged. Look at what the very last protein that's in that inner membrane is called. ATP synthase. To synthesize something means to make it. So this is an enzyme, ASE. ATP synthase makes ATP. It lives in the membrane and it uses the energy that's in all of these, the energy that's left behind by those electrons in order to power the pump, so to speak. So this is the way this happens. It's kind of like a series of enzymes in a row, like a bucket brigade. So this enzyme accepts the electrons and it pumps hydrogen over here, and then it moves those electrons to the next enzyme, which is going to pump some hydrogens in, and then it moves it to the next one. Not every one of them pumps hydrogens, but 3 of the 5 do. So since some of their energy is being used to pump the protons or the hydrogens with, only proton across the membrane, those electrons are going to decrease in energy as they come along, but there's still going to be a little bit of energy in them. So we'll talk about that in just a second. This kind of shows what happens. It comes in here. The NADH drops off its electrons. The FAD drops off its electrons. Protons are pumped across. Those hydrogen protons are pumped across. They build up over here. So they take those high energy electrons. They pump the protons up across. The protons build up in that space in between the 2 membranes. Something's got to give. You can only hold so many protons in that little tiny space in between the inner and the outer one. Well, those protons are going to have a way to get out, and they do so through the ATP synthase. Notice you got ADP coming in, it's becoming ATP. Let me show you how that happens. So, yeah, I'm gonna skip these and talk about this. Okay, so this is the ATP synthase. This is what we call complex 5. We have those first 4 complexes are pumping protons into that inner membrane space right there where they pile up. They get more and more and more concentrated. Now those proteins want to get out, and they can do it through this ATP synthase. They flow down the concentration gradient. So they flow from where they're highly concentrated to where they're not. And as they do so, they literally cause that protein to spin like a water wheel. That ATP synthase in those membranes spins around, and that energy that's generated by that spinning converts ADP, adenosine diphosphate, to ATP, adenosine triphosphate. So here's an animation that shows it happening. So you've got your FAD and your NADH coming up. The energy, the electrons stay in the membrane, being passed from one of these complexes or one of these proteins to another. The hydrogens are piling up over here. They got to have a way out. They do that with the ATP synthase. They cause it to spin like a water wheel, and that converts that ADP into ATP. All right, so let's look at one more thing in this. So at complex 4 right here, in fact, let me take a look at this right here. At complex 4, we have oxygen. See this O2 right here coming in? Got 4 hydrogens and an O2. I want to now keep that in mind as we go to this. So at complex 4, we have that oxygen. This is really the biggest reason that we breathe in oxygen, because oxygen is going to accept the final electron. So remember I said the electrons move along, move along, move along. What then? What has to happen to them then? They've got to be, something has to be, that we have to deal with them in some way. Remember, they're losing energy as they're moving from one protein in the membrane to another. but they still have a lot of energy in them. They still have enough that it wouldn't be safe to leave them in there. They would do some damage. So those electrons are picked up by oxygen molecules. And together with the protons that were pumped out here, that came through that ATP synthase, they are converted into an H2O molecule. There isn't any safer way to get rid of the extra electrons that aren't needed. We make water out of them. It's the safest thing there is. So carrying away the electron in the form of water is the role of oxygen in the electron transport chain. But it's really important, because if it wasn't for that oxygen coming in and carrying away those electrons and those protons, those electrons would build up. They would build up in the membrane. The energy from them would destroy the cell. So the oxygen and hydrogen protons form water in order to prevent that. And that's really critical for the cell to be able to live. Another reason that we have to pick up those extra electrons is if we didn't, the whole system would grind to a halt. You really can't just have piles of electrons hanging around. Remember that this electron transport chain, these proteins embedded in the membrane, they also are going to convert that NADH back to NAD. They're going to convert that FADH2 back to FAD. Those two are then going to go back to glycolysis and go back to the citric acid cycle. Well, if the electron transport chain came to a stop, so would the production of NAD and FAD. You wouldn't have them made to be able to go back to the beginning and start the whole thing all over again. That's what happens when there isn't any oxygen. So that oxygen has to carry those electrons away. And it does so by combining with hydrogen in order to form water. All right. Remember, we said a working muscle cell uses about 10 million molecules of ATP every second. Now, we have somewhere between 1 and 2,000 mitochondria in each cell to keep up with that demand. But think about the math that's involved here. We're using 10 million every second. So each one of those, let's say 1,000 ATPs, has to produce 10,000 ATP molecules every second. I'm sorry, those 1,000 mitochondria. So we use 10 million a second. Every one of those 1,000 mitochondria has to produce 10,000 ATP molecules every second. You can see that mitochondria are pretty important. They are hard workers. They're constantly at work. And some cells are going to have way more ATP than others. Your bone cells are not going to have as many ATP as your muscle cells. That makes sense. They don't need as much energy. But somewhere between 1 and 2,000 are found in every cell that you have. So look at that word, oxidative phosphorylation. So I used that term at the very beginning. Basically, the enzymes themselves that are embedded in that membrane, that inner membrane, those are called the electron transport chain. The process in which this whole thing happens is called oxidative phosphorylation. So oxidative refers to oxygen. It refers to something being chemically combined with oxygen. We know that it is the hydrogens and the electrons that are being chemically combined to make water. The word phosphorylation is a chemical process that involves adding phosphate to something. Remember ATP, adenosine triphosphate. So we're taking ADP, adenosine diphosphate, and adding a phosphate. So that's why it's called oxidative phosphorylation. We're combining it chemically with oxygen, and we're adding that phosphate group to the ADP, oxidative phosphorylation. So right here, you can see the whole thing right there. All right, so when do we use these different kinds of respiration? Well, the phosphagen and the glycogen lactic acid, those are both anaerobic. So we don't really use those very much. We want to use oxygen when we can, because those processes, phosphagen system and the lactic acid, they don't give us a lot of ATP. They give us a little bit. They're going to give us that burst that we need, but it doesn't last for very long. Now, aerobic respiration, that whole process that goes through glycolysis, citric acid cycle, oxidative phosphorylation, that gives us a lot of oxygen. So we get 2 from the first step, that is the glycolysis. We get about 2 from the second step, which is the citric acid cycle or the Krebs cycle. We get about 36 from the electron transport chain. That's a lot. Now, that can last for an unlimited amount of time as long as we use it up slowly enough to allow cellular respiration to recycle it. It's when you are using your muscles faster than, you're using your ATP faster than your cells can regenerate it, that you start getting fatigued, you start getting tired. All right, so remembering enzymes now. Those are the proteins that catalyze chemical reactions so they happen fast enough. The breakdown occurs, so this is what we call a metabolic pathway. This is a series of reactions in which the reactants or the products of one become the reactants of the next. They just go on and on and on through. So in a metabolic pathway, like the one where we have glucose, glucose breaks down gradually by moving through these various metabolic pathways. So how does the cell get the energy out of ATP? Enzymes. How does ADP become ATP again? Enzymes, right? This is really, really important. There are about 75,000 enzymes that are thought to exist in the human body. 75,000 enzymes. And we know that they perform at least 10,000 different functions. So we divide them up into a couple of different categories. We have metabolic enzymes, we have digestive enzymes, and then we have food enzymes that would come in the food that we eat. Enzymes are really, really important. So enzymes are so important that they have to be regulated. They're regulated by other molecules that will either increase or decrease their activity. So these are called enzyme inhibitors or enzyme activators. They're pretty important too. Notice that the activators are often other enzymes, and the inhibitors are often other enzymes as well. Okay, let me ask you a couple questions in the minutes that we have left. A fever of over 105.8 will begin to destroy enzymes in the body. What will be the result? What do you think is going to happen? Enzymes work best when your body temperature is normal, right about 98.6 degrees. Yeah, the reactions are going to slow down, right? Because as the temperature increases, enzyme reaction increases too. You get warmer, you get a little bit of a fever, your enzyme activity increases. But if the temperature goes too high, the enzyme stops working. So that's why you may end up with an inefficient system, and you can have a breakdown. It can disrupt some of your body's functions here. Because they lose their mitochondria during development, red blood cells cannot perform aerobic respiration, but they do perform glycolysis in the cytoplasm. What would happen if glycolysis were blocked in a red blood cell? There wouldn't be any energy. If glycolysis was blocked, so we talked before about how red blood cells lose their nucleus. They also lose their mitochondria. So they have to rely on just a little bit of energy, but they don't need a lot of energy. Red blood cells don't really need much energy. They're pushed along by the blood as it moves, and the oxygen and carbon dioxide leave them and enter them because of passive transport. Energy is not needed in order to do that. All right. You guys ever heard the term rigor mortis? So this is a Latin phrase that means the stiffness of death. So rigor mortis is the stiffness of skeletal muscles after you die. So what causes it? Well, understand when you die, at the time of death, stimulation of the muscle cells stops. But some of those muscle fibers might have been in mid-contraction at the time of death. So you might have had the myosin-actin cross bridges still intact. Now, since the last of the cell's ATP supply is used up at the time that it dies, many of those cross bridges stay contracted. It takes ATP to uncontract it, if you will. So the muscles in that body might be stiff just because those individual muscle fibers ran out of ATP before they could turn off the contraction. Rigor mortis usually starts within an hour or two after you die, and it can continue up to about 12 hours afterwards. And then it gradually decreases, and the body will gradually go back to back to normal. So with that, we're out of time. Anybody have any questions? I know this was a lot today. All right. If you're not, I hope you guys all have a wonderful Thanksgiving. Again, remember that we do not have classes all next week, but you do still have things due today, tomorrow and Friday. So don't stop working now. Thank you very much. Leave this class with a really, just an appreciation for everything that your cells are going through every minute of every day, every second of every day. It's just amazing. When you're giving thanks at Thanksgiving, give thanks for your amazing human body. All right. Have a great week, everybody. I'll see you in a couple weeks. Bye.